Thanks very much, Brennan, and thank you very much for the invitation to be here today. I'm delighted to have been invited to join this conference, and I've thoroughly enjoyed it so far. It's been really stimulating and a beautiful place to have a meeting. Thank you once again for the opportunity to be here, and I'm delighted to be talking about a topic which has been close to my heart for many years, which is enzymes, and particularly enzymes for milk. I guess we are taking a bit of a change from yesterday's particular emphasis, I guess, on microbiology to talk about something a little bit different. So for those who may be less familiar with what exactly enzymes are, I guess enzymes very simply are biological catalysts. They're things which act on one particular molecule, like what we call a substrate, and change it into a product. Their proteins themselves, their activity and their behavior is dependent on factors like pH, the medium they find themselves in, the temperature which they're exposed to, and as we'll see, they can be activated, inactivated, and their activity modulated in different ways. And they're hugely important in a whole vast range of biological and food-related phenomena. I mean, I just take one example here of lactose hydrolysis, for example. We can see lactose hydrolyzed milk where we add an enzyme called beta-galactosidase. We can immobilize it in a simple system, and it breaks down a lactose to glucose and galactose. I'd start with, in terms of their relevance to milk and cheese, I'd start with maybe a bold system. We heard a lot about microbiology and the role of microbes and the importance of microbes in cheese and milk yesterday, but I'd start with kind of countering that by saying that no enzymes, no cheese. And cheese wouldn't exist without enzymes. Enzymes to the milk, enzymes to the coagulant, enzymes to the microbes. That cheese is a hugely, as well as having a vast, as we saw yesterday, and we're learning more about it every day, the extent of the diversity and the complexity and the microbiology of cheese. So cheese also contains a vast range of different enzymes, which contribute to every aspect, from the coagulation of the milk, to the ripening of the cheese, the development of flavor, the texture, and overall what you might, the cheesiness. There's no enzymes, there's no cheese. I'm just going to, there's some kind of debate about how we refer to enzymes that I'm just going to kind of introduce the terminology that I'm going to be using. Like I'd say in terms of cheese, there are 3 different classes of enzymes which are important. The one that I'm maybe going to focus most on is what I would refer to anyway as the indigenous enzymes. And these are the enzymes which are in the milk and of the milk. So if we find them in the milk, it's because they were produced by the cow. So plasmin, lipase, alkaline phosphatase, lactoperoxidase. They're all, in the case of cow, bovine enzymes, or sheep or goat or human, whatever system. We also find another family of enzymes which we'll refer to as endogenous enzymes, which are found in the milk but aren't produced by the animal that's producing the milk. So these typically result from microbes that have grown in the milk and released their enzymes. I guess the classic example being Pseudomonas. We heard a lot about Pseudomonas yesterday. And Pseudomonas is very interesting from a microbiology viewpoint because from a processing perspective, Pseudomonas is a very fragile, weedy kind of a microbe. It's very easy to inactivate it by pasteurization, but it has the very peculiar property that it produces a protease and a lipase that while the parent bacterium is very easy to kill, the enzymes are incredibly tough. And even if we UHT treat milk, we're still going to find their activity. So Pseudomonas growth, the point about Pseudomonas is it's easy to control it and well, not easy, it's important to control it, we know how to control it, but it's not just whether the bacterium is present or not, it's whether it's had a chance to grow, produce, secrete, and leave behind these incredibly tough enzymes in the milk. And then I guess the third category of enzymes we refer to as exogenous. And these are enzymes which are found in the milk or cheese, but which we've deliberately added in. So they're not, if you like, accidental contaminants or accidental residues like from Pseudomonas growing in the milk, but something we've deliberately added in. I guess the classic example of that for cheese is chymosin, is rennet or whatever coagulant we're adding. And there is quite a bit of controversy as journal editor and reviewer. I regularly have arguments with people about what's the difference between endogenous and indigenous, but this is pretty much the terminology we're sticking to. I organised the first international conference on indigenous enzymes in milk in 2005, and I guess we're trying to encourage this kind of classification system. So if we go into cheese ripening, without going into the detail of what this shows, I guess people are familiar with the basic principles, but I mean, we start off with our main 3 constituents which transfer from the milk into cheese. We've got our casein, we've got our triglycerides from our fat, and we've got our lactose. And what cheese ripening is essentially is a hugely, vastly complex series of pathways to break these down and ultimately yield the things that we know. We start off with fresh cheese curd. It's very bland, it's very flavourless, and over time it develops its characteristics, textures and flavours, and the engine of change for delivering these characteristics of the cheese. And a lot of difference between varieties is the difference between the key active agents, whether they're produced by microbes or by the milk or whatever it is, are these enzymes. So if we take proteolysis, for example, and this is probably the one I've studied most intensively over the years, we start off with our intact caseins, enzymes from the coagulant, also enzymes from the milk, particularly plasmin, which I'll talk about a little bit more later, break these down, cleave various bonds between amino acids to give us intermediate-sized peptides. These particularly are picked up by starter proteinases to break them down to shorter peptides. Peptidases, again, frequently from the starter or other microbes present, break down to free amino acids, which are ultimately catabolized. And in the early stages of this pathway, we see the softening of texture, the changes in texture, and in the later stages, we have more and more impact on flavor and contribution to flavor. So, I mean, there was a very famous, one of my kind of favorite experiments that was done in this in the literature was in Wageningen in the 1970s by a guy called Visser, and he did a really elegant experiment to show how important proteolysis to different enzymes are. And he came up with a really clever way of isolating the different fractions, and he was measuring basically, without getting too much into the detail, this is soluble nitrogen. So we know that casein starts off insoluble. As a result of proteolysis, the breakdown products are more soluble, so measuring the level of soluble nitrogen is a very quick, or certainly a kind of a top-level way of looking at proteolysis. And he managed to come up with 4 systems. One where you had all the enzymes present. We can see over time that ripening time is running in this direction. We needn't worry about the difference between the 2 panels, just different ways of expressing it. But we can see over time, over 6 months, this is in Gouda cheese, he was getting lots of breakdown of the protein, up to 30% of the protein being solubilized in 6 months. When he made a system that had no starter in it, we could see that we had knocked out a significant proportion of the contribution to proteolysis. If we took a system which had starter but no rennet, it had a much greater effect. And again, he had very clever ways of managing to isolate one or other of these effects. And he had a system then which had no starter, which was an aseptic system, no starter, no rennet, and he could still see breakdown over time of the protein, showing the contributions. You can see here roughly the relative contributions of milk enzymes, starter enzymes, and coagulant enzymes to the breakdown. So if we just start with milk, I mean, what enzymes are present? Milk is a hugely, hugely complex enzyme system. It's funny, I always take a joke over the years. We've heard, a real kind of buzzword in science has been omics. We hear about, proteome and the genome and the metabolome. I think that they've missed a kind of an obvious one, which is an enzome, which is all the enzymes in a complex system. And I think milk is a great example of an enzome, because there's probably around 70 enzymes which have been reported since the first one in the 1880s, which was, interestingly enough, lactoperoxidase. And quite a lot of these enzymes are a bit like the Loch Ness Monster. They've only been reported fleetingly in a kind of a slightly questionable sighting, but about 20 of them have been studied in detail, and there's a list of some of them. I'm not going to dwell on all of them because they're not relevant, but I'll pick up a couple of interesting facts about some of them. I think there's an ironic situation, which I always draw attention to when I talk about milk enzymes, which is when I organised that conference I mentioned on indigenous enzymes of milk, we had about 120 people there from all over the world who were the people who are interested in milk enzymes. I'd say if we'd had that conference on lactococcal enzymes, we probably would have had 4 or 5 times the number of people present. We probably know more, have defined more, understand far more about the enzymes of the bacteria we add to cheese than we actually do about the raw material, which is always kind of mysterious to me, that the raw material is still pretty much terra incognita. Maybe it's because I guess there's commercial interest in starter cultures and in developing the best starter cultures, whereas milk is almost taken for granted. It's just something that's always there in the background. But there's no doubt that we know a hell of a lot more, and there are a hell of a lot more people interested in this species here than there is in this species here. And that is not to say, hopefully, as I'll convince you over the next few minutes, that we can just discount this as not being significant. So where do the enzymes in milk come from? Milk is a very complex biological system, and its secretion is very complex, and as a result, enzymes come from all over the place. Blood plasma, secretory cell, the mammary cell cytoplasm, the milk fat globule membrane, which is a very rich and unique source of a lot of enzymes, and white blood cells, the somatic cells. And that's a particular area I've done a lot of work on over the years. We heard about mastitis and cell counts yesterday, the relationship between cell counts and proteolysis. These are really potent sources of lots of enzymes, proteases in particular. Why are they there? There's no doubt some of them are leakage products from the blood. I mean, milk contains, I'll talk about plasmin in a while, but plasmin is effectively the enzyme which is critically involved in blood clotting cascade, or the dissolution of blood clots in the body. It's an entire system, carefully regulated system found in blood, which is critical to our life, which happens to be found lock, stock and barrel in milk. Whether it's a leakage product, or whether it's a phenomenon of the way in which milk is produced, whether it's there deliberately. And, the question is, are they there by accident, or are they there for biological function? There's some very interesting work which has been done in recent years, in particular by groups in Israel, which suggested proteolysis of peptides in milk. They found that proteolysis in milk by plasmin, this blood enzyme I mentioned, can basically produce a peptide which controls milk secretion. That they could basically take this peptide, purify it, inject it into cows, and basically turn off milk secretion. So it looked like there was a feedback loop influencing lactation, which was due to proteolysis. So that really suggests a fundamental biological role for these things, as well as a physiological or technological role. And they're very complex, as we'll see in a minute, in terms of their regulation and their control. A key point is that enzyme activity in milk is not static and is influenced by a lot of different factors. Lactation, in particular, seasonality, diet, health, mastitis has a huge impact on milk enzyme profile, processing, and many other factors. So again, going back to this point of whether it's an overlooked topic or not, we can't ignore it because it's not background. It's not just something that's there and unchanging. It's something which is hugely variable and has to be a source of variation when we make products like cheese from it. So what's the significance of milk enzymes? I guess what are the main reasons we're interested in them? We could talk about deterioration of quality or sometimes contribution to flavour. I always have a particular, again as an editor and a reviewer, I often see that enzymes in milk degrade milk proteins. And I always think, that's that makes it sound nasty. And I always change it to hydrolyze. It's like prejudicial that it's a degrading effect. And I think, I have to stick up for their rights. But like what might be negative, for example, plasmin might have is thought, and there's a lot of evidence to suggest it's involved in phenomena like gelation, H-gelation UHT milk, which should be a negative thing. But on the other hand, it does no doubt it positively contributes to cheese ripening. It doesn't directly lead to flavour, but it's involved in the first few steps of breakdown of the proteins, producing the products which later on, several steps and several enzymes later, will give us flavour. So what might be a negative impactor on one product could be a very positive impact on another. Lipase similarly involved in cheese flavour, perhaps other enzymes like cathepsins and somatic cells. Indices of thermal history. A lot of our interest in enzymes, and a lot of the focus and research has been about using them as indicators for other things. For example, mastitis are the things we can rapidly test for to tell us if there's mastitis, like nagase, or things we can rapidly use to tell us about the processing history of milk. The classic example being alkaline phosphatases, an index of pasteurization, which I'll come back to in a minute. Some enzymes have got antimicrobial activity, lactoperoxidase, anti-inoxidoreductase, lysozyme. So there is a natural protective function in milk. It's one of the great ironies of milk is that when we pasteurize it, we actually make milk a better material for bacteria to grow in by pasteurizing it, largely because we're knocking out a lot of the innate defense mechanisms. The raw milk is actually a more bacterially resistant material than pasteurized milk is. And again, we're interested in the variability due to all the factors that I mentioned. And I mean, one of the things is enzymes, we have to think they act in lots of different ways. The key part is most enzymes, milk enzymes, optimum temperature is obviously going to be 37 degrees. When's it 37 degrees? In the udder before the cow is milked. We will never find, like plasmin, which breaks down beta-casein to gamma-caseins, you will never find a raw milk sample straight from a cow that doesn't have gamma-caseins. That hasn't, by the time, no matter what we do, by the time we get our hands on the milk, enzymes have already acted. The process has already started under the optimum conditions, which is in the udder. It's the optimum, so no matter what we do, enzymes are going to have their way with the milk before we get our hands on Obviously, we then take it, we cool it, we reduce the temperature, which makes it less favourable for the enzymes to act. We maybe dilute out. And the interesting question is, okay, if you have variability from cow to cow and herd to herd, the more you bulk it, the more do you dilute and average out the variations? It's an interesting question. And then we take it into products, like cheese, obviously. And then in that way, but it's interesting because we're subjecting these biological systems to stresses they're never intended to encounter. We're fractionating them, we're separating enzymes and substrates, or we're taking them and pushing them closer together. We're separating enzymes from inhibitors of the enzymes. We're subjecting them to challenges like heat treatment, pasteurization, if we're making pasteurized milk cheese. We're changing the conditions, we're adding salt, we're reducing the pH, we're changing the temperature. So at the end of the day, what their activity, the environment in which they're acting down here is probably going to be fundamentally different to that in which they're optimally intended to act. So the enzyme I've studied most, I guess, over the years has been plasmin, which is this blood clotting enzyme. It's an alkaline protease, which is closely associated with the casein micelles in milk, and it principally hydrolyzes beta-casein to these gamma-caseins and proteose peptones. It also hydrolyzes alpha-S2 casein. So you will never find milk in which plasmin is not present and has not already started to act. And it's a very complex system. As you might kind of expect, perhaps, for an enzyme that's involved in blood clotting, we don't want something like that to act in an unpredictable or uncontrollable fashion. So there's a really tight biological control mechanism, which is entirely found transposed into milk as well. In that we have an inactive form, plasminogen, which is converted to plasmin by a series of enzymes, other enzymes called plasminogen activators. We have other enzymes or molecules which act as inhibitors of plasminogen activators, and we have other substances, including beta-lactoglobulin, which can act as inhibitors of plasmin. So a very complex system which works very well until we do things like heat it or make cheese from it. And in particular, one of the things we do when we heat or make cheese is we tend to remove or inactivate a lot of the inhibitors. The enzyme itself is very heat stable and very resistant and stays with the casein. But a lot of things we do in cheese, actually, although we go away from its pH optimum, we actually favour the activity of the enzyme by getting rid of a lot of what are the decelerating factors. I mean, if we pasteurize milk, we actually increase the activity of this enzyme just by pasteurizing it. Not because we make more of the enzyme, but because we knock out some of the factors which are inhibiting or slowing it down. So it's, again, an example of how it's very complex and influenced by a lot of factors. And, there's lots of studies out there, but it's very well correlated to things like somatic cell count. We can see here the levels of plasmin and of the inactive form plasminogen. And if we look at lactation, it's well known that its activity increases over a lactation cycle, particularly in late lactation, which in countries like Ireland, where we have a very seasonal milk production pattern, is not an inconsequential phenomenon. So what does it do in cheese? It's very important. It's very important for cheese from a number of different perspectives. Before we make the cheese, it has hydrolyzed casein. So we do get losses, we do get impacts on yield because there's more of the peptides are likely to be lost in the way. But once it gets into the cheese, it contributes to cheese ripening, which is positive, and it's critical for it to act alongside chymosin to break down the casein. As I said, variability in milk adds to variability in the cheese, and it depends hugely, and we've done a lot of work, and others have done a lot of work in understanding how cheese-making parameters influence it. And certainly cooking temperature, for example, in Swiss cheese, again, we can enhance that, or kind of counterintuitively, a lot of heat treatments tend to strengthen and increase the activity of the enzyme. But then if you've got a huge variation in pH in the cheese, if you've got a cheese where you've got a surface or a rind which has got a very high pH, for example, you're going to favour plasmin activity. And sometimes you can see completely different rates of proteolysis between the rind and the relative. If you think whereas plasmin will prefer to act at a high pH, obviously the coagulant prefers to act at a low pH. So if you get a cheese with zones of pH between the surface and the core, you're going to get very different patterns of proteolysis because you're upregulating or downregulating these two key enzymes in the different zones, which is another thing to be conscious of. So we've done work over the years on what it does, what happens if we chew it up, and these are just some basic results where we did small-scale experiments, and we could show that just by adding plasmin, different levels of plasmin to the milk, incubating it to allow it to chew it up, and then it was Formograph, I think we used at the time, you reduce the yield that occurred, you increase interestingly, you can reduce the rennet coagulation time by limited proteolysis, which is, we had some theories about, but I don't have time to go into, and then more extensive proteolysis will increase the rennet coagulation time, reduce curd firmness, etc. So it does have a demonstrable relationship with cheese-making properties in your milk. Just moving on to other milk enzymes, alkaline phosphatase, obviously we've mentioned, its key interest is not really cheese ripening. It might have a small role in dephosphorylating peptides as part of a proteolysis pathway, but its main interest to us, and probably one of the reasons it's one of the best studied enzymes, is because it happens to have thermal inactivation kinetics, which are very close to Mycobacterium tuberculosis. So instead of having to do a complex and hazardous testing for microbiology to determine whether our milk has been pasteurized, we can just test for this enzyme. It's always present in milk. If it's pasteurized, it will be gone. And it's a very simple, rapid test. So, this is one of the examples of how an enzyme could be a really useful thing. Lactoperoxidase is another enzyme people might have heard about. There's been a lot of talk about. And it's an antimicrobial system. On its own, it's not hugely effective in milk, but by adding certain things to it, hydrogen peroxide or other activators, not things which we would instinctively think, of course, are nice things to add, we can enhance the activity. In certain countries where there isn't cold chain, certain countries in Asia, it can be very common to add hydrogen peroxide to the milk to feed lactoperoxidase and preserve the milk. For short-term warm storage. And there have been some studies on cheese ripening. Here was a study in Gouda cheese where they added sodium thiocyanate, and they found a significant reduction in coliforms of coagulase negative staphylococci in the cheese during ripening due to activation of the lactoperoxidase system. So it does transfer into milk, and there is some interest in using it as a safety enhancing measure. It did have impacts on flavour and lipolysis as well. Which leads us on to lipase, lipoprotein lipase, again a blood-derived enzyme, which acts like one of the amazing things about milk is if you look at the type of fat and the amount of fat that's present and the amount of lipoprotein lipase that's present, the calculations have been done to say that milk should be completely rancid within a matter of minutes. And the only reason it isn't is because the milk fat is very well protected by the natural barrier, the milk fat globule membrane, which keeps the enzyme away from its substrate and prevents the rancidity from happening. But milk contains considerable lipase activity. Much of it's lost in pasteurization, but not all. We need to go to higher conditions than traditional HDSD to inactivate it. And it is no doubt involved in the breakdown of triglycerides during cheese ripening and contributing to cheese flavour in this way. So lipase from milk, again, important in cheese ripening. Just to mention, and we don't have time to, I guess, talk about all the enzymes. That we could talk for hours. I could talk for hours more just about milk enzymes. But if we add in coagulants and other things, it could get even longer than that. So just going to mention briefly. Obviously, there's the other things we've mentioned: chymosin and coagulant as a parallel source, working in partnership, if you like, accidental partnership, perhaps with plasmin in the early stages of cheese ripening. But obviously coagulant is a huge source of enzyme. It's our main exogenous enzyme, whether it's calf chymosin, traditional rennet, fermentation-produced chymosin, microbial rennets, new coagulants. We've been involved in quite a bit of work on camel chymosin. There's been quite a bit of interest in using camel chymosin as an alternative. And I guess the key considerations in selecting what coagulant is used for milk are, I suppose, two things. One is we wanted to do two things to coagulant. We wanted, as the name suggests, we wanted to coagulate milk. But we also wanted to contribute to proteolysis. But we need to get the balance right, because if it's too proteolytic, then before it coagulates, we want the coagulation reaction to be much, much faster than the proteolysis. Because otherwise, if it was the other way around, by the time the milk had gelled, a lot of the protein had been broken down and peptides would be lost in the way, and we'd reduce our yield. So we wanted to give us a curd quickly, and then retain enough of it so that slowly over the coming weeks and months it contributes to proteolysis. And sometimes get those wrong, like pepsin, for example, doesn't have the right ratio. And if we use pepsin, we're going to get a lot more proteolysis relative to coagulation. So the activity during ripening and flavour, the cost, obviously labelling implications of the source of your enzyme, heat stability and whey. I talked yesterday about whey processing, and we have to forget, obviously we can't forget a lot of this enzyme gets lost in the whey. And depending on what we want to do with the whey, is that a problem? Not necessarily a problem, but are we going to have to inactivate it if we're going to add it into another product? Having an active enzyme hanging around there isn't necessarily a good thing. So it's not just for coagulation. And also we have to consider side activities in preparations in different types of coagulants, and rennet paste and things often are much more heterogeneous and less pure form than, say, a fermentation-produced chymosin. We've got proteases, we can have lipases and other enzymes as well. So the coagulant, the choice of it, this is a very quick reference, but just to note, obviously, it's very important. Microbes, again, we could talk for hours about the microbiology. I mean, we talked yesterday about microbiology in huge detail, but there's very little reference to the enzymes that these things are contributing. And all of these microbes are contributing a huge number of enzymes. Lipases, proteases, enzymes which contribute to lactose breakdown and metabolism. And again, where do they come from? Microbiology, they can come from anywhere, from the milk contaminants, like we talked about Pseudomonas already, cheese contaminants like non-starter lactic acid bacteria, or even marine bacteria, as we heard about yesterday, or added cultures. Obviously the starter cultures, but also the molds or the other things we might add, variety dependent. So there's a huge complexity of the microfloras we heard yesterday, means a huge potential complexity of the enzyme system present. And again, you have to take into account how does that relate to things like the pH of the cheese, the other conditions, and whether that favours or discourages the activity of specific enzymes. Some predictable and controllable. We know very well, obviously, our lactic acid bacteria. We know what they do. We know what their enzymes are. As I said, they've been extensively studied. So there's a certain predictability about that. But then you've got all of these bugs that we're still discovering using the techniques we heard about yesterday. We're discovering every day new bugs we didn't even know were present in cheese. So how can we predict what their enzymes are going to be doing? So, and again, in specific cheese types, like mold proteases are critical in mold ripening cheeses, metalloproteases from Penicillium and others. And again, we could talk, like even Lactococcus's proteolytic system is vastly complicated. We've intracellular enzymes, we've extracellular enzymes. Some don't play a role until the cells die and lyse and release the enzymes. Some are metabolized inside the cell, some are metabolized outside the cell. There's a zoo of enzymes responsible. So again, you know, this isn't perhaps very helpful, but it's illustrating the complexity and the key role, again, that enzymes play. So the microbes are hugely critical, but what they do, a lot of what they contribute and how they contribute and influence flavour and characteristics is through the roles of these enzymes that they possess. Just to finish, maybe a last point that I just wanted to mention is manipulating enzymes. If enzymes are so important in ripening, how can we manipulate them? Obviously, we talked about one key manipulation step to up-regulate or down-regulate activity is pasteurization, but there's others. I mean, what's the main variable that affects enzyme activity besides pH? Which isn't something for most cheeses we can just casually change the pH without changing its characteristic, but one of the main controllable variables is temperature. And you know, there's been a lot of interest over the years. Okay, cheese ripening takes time, it costs money, it's a long, slow process. Can we just accelerate it by knocking up the temperature? And there's been a lot of studies, but never to a greatly conclusive degree, to the best of my understanding, that, you know, if we increase it by, you know, if we ripen cheddar at 15 or 20 degrees or whatever it may be, we will get the same flavour in a shorter time. But there has been interest, and an area, just to mention, that we've done a lot of work on in Cork, is other alternative kind of physical parameters to temperature, and one is pressure. And there was a report in Japan, there was a patent that came out in Japan in the 1990s, a famous patent, that said that if you took fresh cheddar curd and put it under pressure, 50 megapascals, which is about 500 times atmospheric pressure in a chamber, for 3 days, fresh cheddar curd, you could get a flavour profile and an amino acid content equivalent to 6-month-old cheddar in 3 days. And that sounded ridiculous and extravagant, and so we did a study in conjunction with Teagasc, with the research centre Paul talked about yesterday, and we published quite a few papers on this, and we found, no, it doesn't work. It doesn't work. But what we did find was, for example, it did introduce us to some very interesting ideas about what pressure could do to cheese, because we have the chambers that we can subject cheese to. And we found, for example, mozzarella cheese, we could really accelerate the development of desirable functional properties like melting and stretchability in pizzas. But one of the things that we've become interested in more recent years is, okay, if it doesn't accelerate cheese ripening, could we do the opposite? If enzymes are susceptible to pressure, and pressure can activate enzymes and microbes, could we use it to stop cheese ripening? Could we take a fast ripening, you know, delicate cheese with a particular optimum window at which it's a perfect flavour, hit it with pressure, and more or less freeze it without freezing it, if you know what I mean, in that state, to give us more distribution and give us more high quality window of consumption? And this has been an area we've been very interested in. We started with camembert cheese, and it works really well. And if we find a market for grey camembert, then I think that's going to be a real commercial success, because unfortunately it inactivates the mold on the surface as well. But what it did work more better on is blue cheese. And we and others published some papers showing that we could basically arrest the ripening of blue cheese using high pressure. So it enhances safety, obviously, and equipment are increasingly available. It used to be a technology, it's really expensive, it's reasonably small scale, and it used to be rather inaccessible technology, but its use is effectively increasing exponentially. The first high pressure unit is opening next month in Ireland as a toll processing operation where people can bring their samples, book time, and just run cycles on the machine. And it's interesting, it might sound like something that's out of the realm of artisans, but a lot of the products which are the breakthrough successes for high pressure, products where there was a unique advantage were things like oysters, guacamole, juices. And a lot of companies in the US and Japan and elsewhere that really made a success of this were small artisanal producers at the start. So I think it's not something to be discounted. So anyway, to finish, enzymes are key to cheese manufacturing ripening. I said at the start, no enzymes, no cheese. There's multiple sources, some of which are controllable, highly controllable, like our coagulant and our starter, some less so. And we can't overlook this variability. And coagulant does a lot more than coagulation. Microbial enzymes had just an insane level of complexity on top of the whole situation. Okay, so I hope that was useful, and thank you for your attention. And I'd be happy to take questions later.